Hello students, Pascal here, and welcome to the 5th video in the series Pymol for Beginners. Today we'll be taking a look at alignment. So when we have two different proteins and we want to see how they align together. This obviously works best when you have two proteins that are very similar or the same protein that's in a slightly different conformation. And that's actually what we'll be looking at today, a change in conformation due to ligand binding or binding with another protein and how that affects the overall shape of the protein that we're interested in. So let's go ahead and launch our Pymol. And we'll be looking at a different PDB code than what we've looked at so far. So today we're going to fetch space 2QCS. And we'll show it as a cartoon. So show space cartoon. We'll hide space lines. And we'll color space. Let's color firebrick. Firebrick is one word. Let's get rid of the waters. If you zoom in, you can see all these little red dots. Those are the waters. So under hide, we can go down to waters and that will get rid of them. Now this is actually two proteins here. One is the catalytic subunit and the other is the regulatory subunit. We're only interested in the regulatory subunit, so we're going to remove completely the catalytic subunit. So to do that, we'll go down to the sequence. The catalytic subunit is the first one. So if you can see here, it says 2QCS. A, so the first one there, A, is the catalytic subunit. So we'll go across selecting all of these residues that correspond to the catalytic subunit. And it's quite big. Oh, I went a little too far, so I'm just going to go back. You can see here it says B, so that's where our regulatory subunit starts. And to get rid of this, we're just going to type remove space, open parentheses, SELE or SELE for selection, close parentheses, and enter, and that gets rid of the catalytic subunit. So now we're only looking at the regulatory subunit. I'm just going to zoom out a bit. And now we'd like to open the other protein, and we're going to take a look at the one that we've looked at in the past. So fetch space 1 RGS, enter, and we're going to show this as well as a cartoon. We'll hide the lines, and then let's color this one deep teal. So color space deep teal, one word, comma, space 1 RGS, so that we add the 1 RGS at the end so that it's only that protein that gets colored. Now, if we want to align these two, we can either align all of the residues together, or we can select specific residues that we'd like to align. So I'll start off by showing you, if we're just aligning the entire protein together, how we would do that. So we'll go here to our 1 RGS, and under A for action, we'll go down to align. We're going to align it to a molecule, and that's the 2QCS. So we'll hit that, and I'm just going to bring this back into our view here, and you can see all of these yellow lines are showing the amino acids that correspond in the two proteins, but how far off they are from each other. So this is not a very good alignment based on how much yellow that we're seeing here. So we'd like to improve that. Now I have a little bit of knowledge of this protein, and I know that part of it will align quite well, and then there's a large conformational change in the other second half of the protein due to the ligand binding in one of these forms. So in order to improve this, we're going to align just the first part of the protein. So I'm going to delete all of these yellow lines. So under the alignment here, I'll go to action and delete. And now let's align just the first few residues. So we're going to go back to the start, and we're going to align residues 113 to 160. So I'm going to highlight these ones, and you can see here our selection. I'm going to rename this so that I can use it to align. So under action, I'll go down to rename selection, and I'm going to name this 1 RGS underscore 113 to 160. So I know what it is that I'm aligning. Now let's do the same thing for the other protein. So we'll go from residue 113 to 160, and again we'll rename this. So under selection, we'll go to action, rename selection, and this time it will be 2QCS underscore 113 to 160, enter. Now let's try aligning only those particular residues and see how that improves things. So we'll go to our 1RGS underscore 113 to 160 action, and we'll go down to align. This time we're going to align to a selection, and we're going to align to that selection of the 2QCS that has the similar residues. So we'll click on that, and we can see that only part of the protein is aligned. This is the domain A, and these two are aligned, and you can see very few of these yellow lines indicating that there's a much better matching. So what's actually happening here is domain A matches very well, but this helix here has a large conformational change upon binding and bends around like this, and then the second domain is in a much different location than in this other protein. So there's a great article if you're interested in what's happening here by the lab of Susan Taylor. It was in Cell 2007 called PKA1 holoenzyme structure reveals a mechanism for cyclic AMP dependent activation. So this is a great paper that explains what's going on here, and they show these same types of diagrams to explain what they're doing. So this is how we do alignments in Pymol. Hopefully this was helpful to you, and we'll see you in the next video. Bye-bye.